It started out in 2016 with a couple of people who were bugging the CDC about different bioinformatic needs and in the state public health labs, and finally CDC people getting tired of hearing the same question over and over from all these state public health bioinformaticians said, hey, why don't you guys form your own group and you know, start working on your problems together because you guys are all working on the same things. And so we did. And we started out with a ragtag team of a few bioinformaticians, and we've been growing ever since. So I wanted to just highlight some of our little accomplishments. So our membership, the last time that I looked at the email list, we have 132 members, which I think is pretty awesome. And 49 different organizations are represented. And so I put together this little map of all of the states that I have representation for in our email list. And if I missed your state, please shoot me an email and let me know, because I would love to fill in this map all the way. I think that it's really awesome that we've been able to grow to the size that we are, and we have such a diverse and wonderful group of members. And in closing, I wanted to point out that we have lots of different resources that are available. We have our private YouTube channel, which is available through the StaPH-B webpage, but we also have our StaPH-B resources are available on the StaPH-B web page, as well as there's the Slack channel to talk to anybody if you have any problems or if you have any questions. And you can get all of those resources either on the StaPH-B website, which is staphb.org, or by contacting any one of the members of StaPH-B. I think anybody can direct you over there. And so I just wanted to say thank you all for being a part of StaPH-B, and I really appreciate you guys. And so with that, I am going to hand over the mic to Rebecca and Richard, who are going to be presenting on ViPR and PATRIC, so two different resources for viral and bacterial analysis. So I'm going to stop sharing my screen, and if you want to take it away, I'll let you guys go for it. Hopefully you have.
So it says host disabled attendee screen sharing.
All right. Sean, do you know?
I just changed it to.
Yeah, now I have access.
Okay,
yes.
Perfect.
All right. So hopefully you are seeing my presentation now. Yeah, so Logan, thanks so much for the invitation to talk to this group. It sounds like it's a pretty interesting community and very relevant for our resources. So Rebecca and I are going to tag team on the presentation. I'm going to start by talking about the virus pathogen resource, which is part of the Bioinformatics Resource Center's program funded by NIAID. And then Rebecca will talk about the PATRIC resource, which is focused on bacterial pathogens. You know, we don't have a whole lot of time today, so we're going to give a very kind of high-level overview of the resources to give people a sense for what's available. And then I'm going to show a couple of specific use cases around SARS-CoV-2, since obviously this is something of great importance to the public health community currently. So the BRC program originated by the Division of Microbiology and Infectious Diseases at NIAID as a way of trying to provide support for their large-scale genomics programs. So they have genome sequencing centers, functional genomics centers, structural biology centers, systems biology centers that are generating a large quantity of data that are useful to the broader infectious disease communities. And so they wanted to have a single place where the data that's being generated by these programs can be housed and disseminated to the broader community. And so that was the goal of the Bioinformatics Resource Center's program. So there are currently 5 kind of bioinformatic resource centers: PATRIC, ViPR, IRD, UPathDB, and VectorBase. We're actually in the process of consolidating these into 2. BVBRC is going to consolidate support for bacterial and viral pathogens, And then ViewPathDB covers eukaryotic pathogens and the vectors of infectious disease. But they're all kind of part of this BRC umbrella program. This is, screenshots of the two viral databases, IRD, the Influenza Research Database, and ViPR. IRD is exclusively focused on influenza, and then ViPR covers support for a pretty broad range of different human viral pathogens and also related pathogens that infect other organisms. The idea behind these resources is to provide this comprehensive integrated database of human virus research and surveillance. The resources are free and open access with no restrictions. You can download the data and do whatever you want with it. And our focus has really been on making sure that the data that we're providing is well curated, that the annotations are consistent, that the data is integrated and aggregated in effective ways. And we also generate novel data through different kinds of data processing and analysis pipelines that would be unique to these resources. So for IRD and, sorry, I'm hearing an echo. For IRD and ViPR, we get about 3,000 usage sessions per week, and they've been cited in close to 1,500 publications. Can somebody mute? Oh, okay, I think that's better. We do get data from a number of different sources. So we aggregate data from GenBank, from UniProt, from the Protein Data Bank, from the Immune Epitope Database. We do reach out to these kind of archives, all of the virus-related data, and then put those into IRD and ViPR for access. We also have direct submission of data sets from particular NIAID programs. So for example, we work closely with the CEERS program, which is Centers of Excellence for Influenza Research and Surveillance. And so all of their surveillance data comes into IRD. So we have over 1 million surveillance records for influenza-related surveillance projects. And then the third source is data that we derive from our own data processing and analysis pipelines. And so one example of that is in the case of SARS-CoV-2 genome sequences, we process those through our own annotation tools so that we provide consistent annotations of all the predicted open reading frames and mature peptides that are generated through proteolytic cleavage of the large polyprotein. We have kind of custom query interfaces that you can then use to retrieve data records from the databases. This is a screenshot of the protein sequence search page for influenza. And just to note some, I think, important unique features, in addition to be able to search for classical proteins for influenza, the hemagglutinins and the neuraminidase and non-structural proteins, NS1 and NS2, we also, the research community has defined a series of variant proteins that are produced from influenza due to alternative splicing and translation, slippage, and other kinds of mechanisms. And so we have algorithms that search for the presence of these variant proteins in the genome sequences as they're getting loaded into IRD. And then we make those variant protein annotations available for search as well. And then obviously the kind of the standard metadata characteristics about, the host, the geographic location, and the time of isolation, all of those can be used as search criteria. So we have the data part, and then we have a whole suite of different analysis and visualization tools that include things like BLAST, phylogenetic tree inferencing, multiple sequence alignments, different kinds of clade and genotype classification tools, a variety of different things to help support comparative genomics analysis and visualization. And then finally, the third major part of the resource is a workbench function where you can set up your own private workspace, and you can, pull out subsets of data records and keep them, in a group for you to do analysis. You can save your analysis results. You can even upload your own private data and integrate that with the data in the IRD and ViPR databases, and then do an integrative analysis of your private data with the public data. And so it's pretty easy to set up your own kind of personal workspace and use the tools and resources in that kind of an environment. This is kind of an interesting graph, and we see this every time there's an outbreak. We see that the usage of these resources, spike pretty dramatically. Here, this is usage of both ViPR and IRD. So prior to the COVID-19 outbreak, as I mentioned, we get about 4,000 to 5,000 users per, I guess this is per month. And when the outbreak occurred, we saw this big spike in usage. But interestingly enough, not only was there a spike in ViPR usage, but there was also a spike in IRD usage, which is interesting. And then things quiet down again, but what we have seen is that this kind of plateau in regular usage has increased by about 50% as a result of the outbreak. So in some ways, these outbreaks, I think, highlight the availability of these kinds of database resources. And once people are aware of them, we see that our usage statistics tend to stay higher. So we've made a couple of specific enhancements related to COVID-2 outbreak. The first one was that in February, we set up a portal that allows easy access to all the COVID-2 data that we accumulate for SARS-CoV-2. We currently have actually over 15,000 genomes in ViPR, and actually, I think, sorry, we have over 30,000 genome sequences in ViPR currently just for COVID-2. And so this portal just makes it pretty easy to access all of the data related to COVID-2 through ViPR. So we established that portal pretty early on. In addition, we have a couple of tools that we developed that were tailored specifically to support COVID-2 research. So one of them is something that I mentioned earlier, is that we have an annotation tool that we've developed over the last couple of decades, actually, called Vigor. And this just allows us to annotate open reading frames in viral genome sequences. And so we kind of enhanced the Vigor 4 tool to be able to support the prediction of all the COVID-2 open reading frames, but also the mature peptides that are generated from proteolysis of the polyprotein. And so on the right-hand side here, this is just an example of a genome annotation report from Vigor that includes, the location of all of the mature peptides and open reading frames, some basic statistics about those encoded proteins, and then you can download all of the information about them and all the sequences. And now this is routinely available for all of the sequences that we have for COVID-2, because we routinely process the genomes as they're coming into ViPR using this annotation tool. And then the third enhancement that we added is now for those people who are actually doing their own sequencing of COVID-2 samples, we have a genome assembly and annotation service that you can use that will take FASTQ files, read files from either the Illumina platform or the Oxford Nanopore platform. And we will assemble those reads into contigs and then process those using the Vigor 4 annotation tool to provide not only the assembled genome sequence, but also all of the annotations that go along with that. And so this is available now as a service. So you can upload your FASTQ files and then download all these different kinds of annotation reports from this workflow. And then finally, the fourth thing that we're working on right now, this is kind of a works in progress, but I think this is going to be pretty interesting just as a general resource for coronaviridae research. So we have a method for calculating protein orthology that incorporates domain architecture information as well as phylogenetic relationships between orthologous proteins, and defines what we call strict ortholog groups based on the sequence similarity, the evolutionary history, and the conserved domain architecture of these proteins. And this schematic here is just an example of the polyprotein for different members of the coronaviridae family. So down here you see the Sarbico group. This includes MERS SARS and SARS-CoV-2. This is the domain structure of the polyprotein. And what you can see is that actually that polyprotein is quite different for different members of the coronaviridae family. The amino terminus seems to be quite variable, whereas the carboxy terminus seems to be quite well conserved. So this is something that we're working on, developing the algorithms to compute these ortholog groups, and then this will be made available in the ViPR resource in the near future. But these are just some examples of the kinds of things that we do in the setting of an outbreak to really enhance the resource to support the specific needs of people who are exploring these outbreak pathogens. So I don't want to take up too much of Rebecca's time, but I'm just going to give you a couple of quick examples of kind of using the resource with a live demo. And feel free to ask questions while I'm kind of getting set up. Okay, so this is the ViPR homepage. It's broken down into different virus families that cover, flaviviruses, coronaviruses, filoviruses, pox viruses, herpes viruses, etc. Whenever there's a kind of a high-profile virus that is getting special attention by the research community, we set up these feature virus portals, and I showed you a screenshot of the SARS-CoV-2 portal. And so those are available for kind of selected classes of viruses. But typically when people kind of come to the resource and want to explore the data and use some of the analysis tools, they start out by coming into a particular virus family. And now here you can choose to search for data, analyze data, and then use this kind of private workbench function. And so just as a quick example, if we choose to do a sequence search, you have the options to search for genomes or proteins or strains. We have here, we kind of keep track in real time of the number of records that would be returned based on the queries that are being constructed. On the left-hand side here, this allows you to search based on the virus taxonomy. And so in this case, if we want to drill down into the beta coronaviruses and then into the SARS coronavirus species, we can choose to basically select all SARS-related coronaviruses as one of our search criteria. And you'll see up here the counts will kind of update in real time. I might want to just focus the analysis on complete genomes, so that's an option here. In this case, we might want to just limit our search to the current year to really focus on SARS-CoV-2, and then maybe even limit the search to only isolates from human. And so now you'll see that as a result of those search criteria, we have about 26,000 records that match those search criteria. And so you can then launch the search. And then what we provide as the results is kind of a summary table. that summarizes the records in the database that match those search criteria. And that can take a little time. And so now you get this kind of summary table of the different records that match those criteria. You can adjust the display of this table. You can choose different columns to display or hide. You can sort the table based on different values in the different columns. You can adjust the number of items displayed per page, etc. So you can kind of customize the display of this table. And then if you kind of drill into the individual database records, this is an example of a strain detail page for this particular virus. There's basic information about the strain. There is information about the genome sequence and the providers of that sequence information. If you want to view the nucleotide sequence of the genome, you can click this link here. And this takes you to the sequence. And one of the things that we really try to do in the resource is integrate the data with the relevant analysis tools to make it very easy to go from a data set to an analysis process. And so, for example, here, when you're at the genome sequence page, you can either choose to download that sequence, or you can go right into PCR primer design. And so we have an implementation of the primer 3 algorithm. And so now here we've loaded the genome sequence into the primer 3 tool. And now you can customize the search for PCR primers to include or exclude certain regions of the genome, have primers of certain sizes with certain melting characteristics or GC content or certain product range. And then have the tool pick primers and all of these kind of criteria for primer design. Another feature of ViPR is up here we have what are called breadcrumbs. So we're basically kind of keeping track of how you're migrating through the system. And this makes it very easy for you to go back to a page that you were several steps prior to where you are currently. So if I go back to the strain detail page here, this is where we were. And now as you scroll down here, here's information about the encoded proteins. And we've divided this up into 2 sections. These are annotations that we get from the GenBank record that is part of that GenBank file. And then, as I mentioned, as we bring these sequences in, we run them through the Vigor4 annotation tool, and we get all of these annotations that kind of go beyond what you might find in the GenBank record. We find the annotations in GenBank are quite variable. In some cases, you have full annotations. In other cases, you don't. But in ViPR, we have consistent annotations for all the sequences. So I'm going to kind of come back and just show one more kind of quick example here. So I'm back to the search page. And what I'm going to do is change this from a genome search to a protein search, and then come down here and look for the RNA-dependent RNA polymerase. So I still have my kind of year and host selections up here. And now, so I'm going to launch this search now. And so what this should do is then return all of the records for the mature peptide encoding the RNA-dependent RNA polymerase. So we have 24,000 of those proteins. But the other thing that I'm going to do here is go back to the search criteria, and now I'm going to add in some country search criteria. And just for the sake of an example, I'm going to select sequences from China, from Italy, and from Spain. Redo the search. Okay, so we have 113 proteins that match those criteria. And I'm going to give one last example here of how we integrate the data with the analysis tools. So if I select all of those 113 protein records and then come up here, I have the option of using a number of different analysis tools that are consistent with the data type that are in my data set here. And so one of the unique tools that we have in ViPR is doing metadata-driven comparative analysis. This is a tool that we developed called Metacats. And so what you can do here is you basically are going to compare groups of sequences to identify regions of dissimilarity between the groups. So you can either generate manual groupings, or you can use this auto grouping function and group the sequences based on these metadata characteristics that are in the database. So in this case, I'll choose to do auto grouping by country. And now if I move forward, you can see now that I have generated these 3 groups of sequences based on the country of isolation. You can verify here that the segregation of the sequences into these groups seems to be correct. These are all from China. These are all from Italy, and these are all from Spain. And then I can choose to run the tool. When you launch a job, you get this page. You can choose to save the results to your workbench directly here. You can choose to receive notification by email if you don't want to wait around, but this happens pretty quickly. And so what this tool has done is it's done a multiple sequence alignment of all of those sequences, and then it has compared each positional sequence alignment using a chi-squared test of independence. And what we found in this analysis is that there is one position that's significantly different in the RNA-dependent RNA polymerase between these groups of sequences. And you can see here that is a summary of the amino acid residues at that position. Group 2 is from China, and 45 out of 48 of the sequences have a proline at that position. But if you look at group 3, for example, from Italy, you see that 32 out of 33 of those sequences have a leucine. And so this has, a p-value of about 10 to the minus 15 based on this chi-squared statistic. But this allows you to very quickly be able to look for, residues that are significantly different between groups of sequences that you might want to explore. So I guess the only thing I'll mention in closing here is that this is a list of all the different analysis tools in ViPR. And I mentioned this Vigor4 genome annotator. So if you have your own SARS-CoV-2 sequence, you can upload it into the annotator and get this complete annotation output. And then I mentioned that there's this genome sequence assembly and annotation service that takes the raw reads from Illumina and Oxford and produces the assembly and annotation reports. And so you can access these tools and all these other tools through this analysis tools landing page. There's a lot more stuff in IRD and ViPR. Hopefully you get a sense for what's available there and how to migrate through the resource. And so with that, I'm going to turn it over to Rebecca so she can show you an overview of PATRIC and the kinds of tools and services that are provided there.
Thanks, Richard. Let's see if I can share my screen. I'll just say share it all. Can you guys see my slides?
Yes.
Okay. I had 2 different talks prepared. I wasn't sure if you guys were more lab biologists or bioinformaticians, but when you put up that little symbol, I knew who I was talking to, so we're going with the bioinformatics talk. So Richard's given you pretty much an overview of what NIH and NIAID want in their bioinformatics resource center program. So I won't go too much into this, but I wanted to point out the kind of data that PATRIC has, like ViPR and IRD. We bring in data from different resources, and a lot of people for a long time thought because we have this drop down showing the pathogens that NIAID was most interested in. They thought that that was the only data that we have. But actually, we bring in all the bacterial and archaeal genomes that are in GenBank, not just the pathogens. And recently, we've started including bacteriophage genomes as well. And the reason we bring this in, all of this data in, is we not only does it have more people using our resource, but also when you're exploring pathogenicity, often you have to make comparisons with things that are not pathogens. So in the data, where do we get this data and what do we do with it when it comes into PATRIC? I see that this thing's on the side. I hope that it's not blocking this fabulous little slide here.
I don't think we see that, Rebecca.
Oh, okay, good. So every month we go into GenBank and bring in new data. We also get data from the genome sequencing centers and also from collaborators. Within the past couple of years, we've also been going into SRA. We look for papers. Primarily, it's been about antimicrobial resistance. Often, as I'm sure you guys know, when you submit a genome to GenBank, there's often a gauntlet that you have to go to submit the data. And we're finding that a lot of people are now just putting, especially with their large studies, they're just dumping them all into the sequence read archive. And so we've started going there. We look for papers because we have some machine modeling learning efforts on antimicrobial resistance. So people are going, we have a dedicated team who's looking for papers and bringing that data too. So when the genomes come in, if they are in GenBank or RefSeq, we keep their primary annotation. But one of the backbones of PATRIC is being able to do these cross analyses. And to do that, we want everything to be annotated consistently. So we keep the primary annotation, but we re-annotate everything using the RAST annotation pipeline in PATRIC, and the RAST team is part of the PATRIC team. Another thing we do that I think is probably one of the most valuable things we do is we specifically mine the metadata. Anybody who's gone into GenBank or SRA, anywhere, and you're trying to find specific data, the searching for that data is a nightmare. So we've been trying to bring out things to make it really easy, or let's say easier, for people to find data. Like in PATRIC, I can quickly go in and find all of the genomes that came from organisms that were isolated from blood or urine or environmental. So we try to do things like that to make it easier for people. You can enter a bioproject number, things like that, as long as it's linked somewhere within biosample, bioproject, or GenBank, or it's mentioned in the GenBank notes for individual genomes. Those are all searchable for us. So the metadata has some manual curation that's tied to the genome and put into the database. When the genome's annotated by RAST, of course, it gets genes and proteins and RNAs, but we also give enzyme commission numbers and Go assignments so that we can map those to KEGG pathways. Most importantly, we have a protein family assignment that we give cross-genus protein families and genus-specific protein families. Many of our tools call on the protein families that help increase the speed. So we're looking for those things. It makes things work a lot faster. And then all of that is in the database. Oops, let's see, where am I? Here I am. So I just started this from 2015. PATRIC started a lot earlier. We started with 8 bacterial genomes, believe it or not. And the data increases. And you notice there's these occasional dips down in the data, what's happening there. Often we find that there's a lot of poor quality public genome data, multiple, tens of thousands of contigs. We try to get rid of those. So we remove them from the database. So what are some of the unique PATRIC features other than the comprehensive data collection and the uniform annotation? One is that we give, like ViPR, a workspace for analysis of now user data. So one of the things we want to have in PATRIC, any genome that comes in, if it's public or if it's private, they've all been annotated exactly the same way so that you can use a direct comparison between those. And we've watched our user numbers increase every year. So what are they doing in PATRIC? Well, they're using these services mainly. We first started in 2015 when we moved more from providing a view of the public data. We decided to go more hardcore on the bioinformatics. So we started with assembly, annotation, model reconstruction, those jobs, those types of services at the top. Then we added in the next year variation service, which is SNP calling, BLAST. And we also, because you guys are bioinformaticians, we now have a command line interface. So you can launch any of these jobs on our interface and use it. You can access the workspace, you can download, you don't have to go through our GUI. Transposon-seq was added in 2017. So each year we've been adding more data. And what we really wanted people to do was to bring their own data into PATRIC and analyze it, comparing it to the cornucopia of public data that's out there. So this just shows what they're doing in the last year. By far, the thing that people do most of all is annotation. That's our major workhorse. I look at every paper that's ever cited PATRIC. Annotation is the primary thing that they're doing. But we have some other stuff that they do quite a bit as well. Some of the harder core, hard things to do in bioinformatics, like assembly. RNA-seq, we have an RNA-seq service. It hasn't taken off as much, but I'm hoping that with ViPR, when we have the human or the host data as well, we'll try to launch this a more broad scale look at that kind of data. So I hope that that'll take off too. So on a per year basis, I just am always trying to figure out what people are doing. Is it a specific thing that we launch that brings things in? I know that this particular thing was someone came in and just annotated tens of thousands of bacteriophage genomes on this 1 month. So that spiked that. But if you look over the years, you can see that a lot of people are submitting a lot of jobs. And this is the average job submitted per year in PATRIC. In 2020, we're not done yet with, so it's just up to the end of September. All of these statistics are up to the end of September in 2020. One thing that has happened, right now we have no limits on the workspace, but the demands on providing the workspace, this is at the Argonne National Lab that's managed by the University of Chicago. The workspace demands are getting pretty intense. So we'll keep it up for free. We don't limit what anybody can put in there. We'll see how long we can keep going as our popularity increases. Each user is the amount that they're using the data or the workspace is increasing along with the jobs that they're submitting. So the size of the individual workspace per user keeps going up, and the number of files that are in the workspaces are going up too. So you can see it's a big database effort that we have to have our public data, our private data, to allow these things to interface so that people can see what they want to see and do the kind of work that they want to do. The thing that's, well, this I think says it all. In 2018, suddenly we went from public data to private data, private genomes in PATRIC being more than the public data that's there. So I think that's a strong indication of how much the resource is being used and that it's growing a lot, and we have a lot of people using it. So PATRIC continues to evolve to meet user needs. We have to update our pipelines. We did the assembly pipeline. Well, Richard described the SARS pipeline, but also for bacterial assembly, we recently updated that. We also updated the phylogenetic tree pipelines. We've started, as we get more into this, we notice that often our users are not, you know, they annotated Genome in PATRIC and then they're like, oh, great, I can go ahead and publish this. But they had no sense of when their data was terrible or when it wasn't. So we've started putting more effort in to trying to let them know when they have a bad genome so that they don't waste their time or they go back and sequence, do another sequence of it. As I mentioned earlier, we have a lot of special data that we bring in, like the antimicrobial resistance data. And then we've started, which I'll show you some of the things, these full-bodied analysis reports that we've started doing for users. So when we first started PATRIC, it was, oh, do your assembly, and then you have to do your annotation, and then you have to do your tree, and then you have to do your analysis of the protein families. And then we realized we could tie a lot of this together for them and give them an analysis that shows them all of that rather than making them step through these individual steps. So what I wanted to do was show you a few of those things that we have in PATRIC. So any questions on that before I launch into everything? We have the services here that make it easy for you to leap in to any of the services that you're interested in. Let's say you want to do an annotation, you can just click on that. You can upload the data here, or if it's in your workspace, it'll show you the list of contigs or whatever it is that you've recently annotated. So we try to make it very easy for users to do these things. But let me show you what some of the jobs look like, especially some of those reports that I was mentioning. So I can click down here and go to my jobs page. And you can even see that I've been pretty active just this morning. I've been working on some stuff for a collaborator who had some genomes that he asked me to annotate and look for specific proteins within them. And during the course of the annotation, I started realizing that he didn't have clean genomes. He didn't have what he thought he had. So I started running different services to try to figure out what was going on. Taxonomic classification service to look to see what was in there. And then I went into metagenomic binning once I realized he had multiple genomes and what he thought was an individual sample. But let's look at this comprehensive genome analysis. And I just want to show you, when you submit a job in PATRIC, the kind of thing that comes down with it. And remember, if you're doing this by command line, you have access to it via command line. You don't have to go through the GUI. So one of the things we always provide, we try to anticipate what people want. Like the biologists are never going to want the JSON files, but the computer scientists always do want them. So we have to provide a lot of stuff with every service that gives people just what they want in different flavors, so that they can take this data and go to other tools or other viewers. You don't have to stay in PATRIC, but we try to anticipate that for things that you'll need to go into other services or other tools. So here's our report that we have. And right away, it tells me if the quality is good or not. It gives me some basic information. So it's going through, gives me a view of the genome, of what we call the subsystems, which are what the metabolic capabilities of this particular organism, did it have any AMR genes or virulence factors or transporters or drug targets? One of the things we like to do is to not reinvent the wheel. And we try to go in and grab other resources that are considered to be gold standard, like VFDB for virulence factors, or also Victors for virulence factors, CARD or NARDO for antibiotic resistance genes, TCDB for transporters, drug bank, and every genome, public or private, that comes into PATRIC, we blast the genes against those to tell people what genes of interest they might have. The antimicrobial resistance genes, NIAID's very interested in that, so we dig deeper in that and try to show them those things. And then we run a quick tree for them to show them if this genome is what they think it is. And then it's on the user to go in and do a more a broader look at that genome if they're interested. Often this is, oh, let me also point out here, is that we also provide them with the citations. I want to make it really easy for people to publish so that they can cite us. And so I even give them words for what they can use in the publication. Like we used MASH, MinHash, and then the global families with that annotation. And then the proteins were aligned with muscle, and RAXML was used. And then they did the fast bootstrapping option. They can take that. That makes it easy for, I think, a biologist or maybe even a bioinformatician, so that you don't have to struggle and figure out exactly what was used in this anonymous pipeline, wherever it was you were using it. I try to make it very easy for them to do that kind of stuff, to give them that. Also, in any of the services that we use, like let's say it's the phylogenetic tree, you can go to the tutorials for that tell you how to do the service. And at the bottom, it also gives you the references for that. Now, because you guys are all bioinformaticians, you might be most interested in the FTP server. So you can go there, or you can access it via command line. We don't keep track of this, but a lot of people just come in and grab all the data, especially if they're bioinformaticians. They're not interested in, they want to do their own stuff. So they just come in because supposedly I'm a biologist, I'm not a bioinformatician, but I know that they tell me it's so much easier than GenBank to grab the data from here at particular taxon levels than it is anywhere else. So that is just sort of the kind of thing I wanted to show you just briefly. Let me quickly go, like you can go to an organism and what Richard was talking about in terms of filtering. So these are all the genomes that we have within the pathogen Brucella. I can click on the filters here and I can see my private data, which is the false versus the true. So I can do a dynamic filter on different organisms. And then I can do whatever I want with this data. I can create groups, and then those groups can be used in any of the downstream tools. So this was just a really fast, rapid demonstration of what there is in PATRIC. And I'm happy to answer any questions if you have any.
All right, thank you both so much to both Richard and Rebecca. This was an amazing presentation, and it looks like a fantastic resource. I probably should have given you 2 hours to give this presentation. It looks like there's so much stuff here that we didn't even, scratch the surface on a lot of these different things. I know I have some questions, but I will open it up to the floor since I know we have only a few minutes left till the top of the hour, and I can stay on for a little bit of time. I don't know, Minnesota folks, if you have a little bit of time extra too, if we have questions that go over, but I figure I'll open the floor for questions first, and if anybody has anything, go for it.
Shall I stop sharing? I can point out here that the command line interface is here, but I'll stop sharing. So,
oh, it looks like we have a question in the chat. Anna wants to know, could I hear a little bit more about metagenomic binning service?
Ah, okay, yes. So, and I just, interestingly enough, I ran a metagenomic binning service this morning. What this service does is it takes, you can start with reads or you can start with contigs. Let's look at the binning thing. So you can start with reads or you can start with contigs. You tell it where you want it to go in your workspace. And then what it's going to do is it's going to try to find good genomes within that, within whatever, and it'll tell you if they're poor quality genomes. So I ran it this morning on this particular sample. So we can go in and look, and I can see now that I started with contigs from this person. Actually, they were reads that I assembled separately. So it created a single bin here, but I can look at the binning report and view that, and it's telling me something about that. It's telling me what genome it looked at, and it gives me coarse and fine consistency, and also completeness and contamination. And moreover, it'll tell me what genes it thinks that what it does is it looks at the reference genome and the reference genomes for this particular genus, and it has an expectation of what genes it should have. And so when you don't see those genes, it will go in and it'll tell you specific problems. It was predicted that this particular genome should have 2 of these genes. It had 3. And then you can dig so deep into this. It's, I mean, you can get lost in this stuff forever. It's sort of like the Matrix, and you can spend the rest of your life digging into exactly what these problems are. But you could see if it was a frame shift or if they're separated on separate contigs. You can look at this one, that this particular one is on one contig, and this particular one is on the second contig. So what I would assume is it's an assembly problem with that. So it gives you that kind of information. Any other questions on binning?
I don't have any questions on binning, but I have some other questions related to some of the things. But just before we get into that, I figure we can probably stop the recording. And I just wanted to let everybody know that the recording will be available on the StaPH-B website later once it's available. And so I will try to send out an email to remind everybody when it is available if you want to go back and look at some of this stuff, because I know that there was a lot that we went over in this video. And so that will be available hopefully later on today or this week.